Hello, welcome to Pharmacognosy and Metabolic Engineering online certification course of NPTEL. So, this is the lecture number 4, that is production of specialized metabolites through cell and organ culture. What we have seen in the previous classes, that how specialized metabolites are synthesized from primary metabolites, and mostly we dealt with that specialized metabolites played important role in the survival of the plant. But we human being are more interested how to exploit these specialized metabolites for our own need, for example, as drugs, as flavors, as fragrance, as aesthetic compounds and so on. So, what we will see now is this, that these specialized metabolites are produced only in specialized plants, and these plants are not abundantly available. These plants are growing in particular regions of the world, maybe particular regions of the country or region, and that too these plants are sometimes short lived plants, they may have only a life cycle of several months. So, in other words, in order to harness the metabolites of interest, we cannot fully dependent on the field harvest for these compounds. So, what is needed basically to look for an alternative system, so that year round production of these metabolites can be maintained. So, how that can be done? So, in order to do that, we need to know the tissue culture, because plant tissue culture is a tool used in biotechnology, where plants are propagated inside the laboratory, and that too specifically inside the test tube. So, these plants are aseptic, that means sterile, and these plants can be grown throughout the year in a particular environmental condition in the laboratory or even in the industry. So, let us now look into the aspect of tissue culture. So, what we are going to cover today is basically the main concept of tissue culture is totipotency, that we will cover. We will talk about cell suspension culture, we will talk about differentiated culture, that is about shoot culture, we will talk about another type of differentiated culture, that is normal root culture. Then we will also briefly mention about hairy root culture, and then we will end this course, this particular lecture, with the empirical methods available to manipulate the level of metabolites in the culture. So, these are some of the specialized metabolites which are produced from plants, and the products are available in the market, and what you see in the right side is basically the finished products, and the different applications are shown here. For example, quinine, antimalarial, digoxin and cardiotonic, thymidine, sweetener, lycopene, red pigment, so on, and the respective plant sources are given in the last column. So, in order to produce these metabolites in plants, what I have mentioned, that we need to go for plant tissue culture. So, plant tissue culture, basically the concept first came from the terminology called totipotency, which was given by a German botanist called Haverland in early 1900 to 1905, in those years. So, what is totipotency? Totipotency is the capability of a single plant cell to grow into a full plant. That means, a single cell has the genetic capability to produce the entire plants, provided you give them the right environmental condition and the right nutrients, including hormones. So, that means, if you can isolate a single cell from the plant, and if you put it in a culture medium, and if you put it the right hormonal combination, it is possible to grow the entire plant from that. So, this is what is genetic totipotency, and in addition to that, there is a chemical totipotency terminology is there. That means, whatever a plant is producing, whatever metabolite, maybe it is producing in root, or it is producing in the shoot or in the leaf, that genetic information is also there in the single cell. So, if we can create a right condition for the single cell to grow and metabolize and means it activates its metabolism. So, it is possible to harness the metabolites of interest from the cells producing from the single cells. So, in other words, cell cultures produced from the single cell may be capable to produce the metabolites of interest. Next point is dedifferentiation. That means, a differentiated tissue, what is the differentiated tissue? Normally root, stem, leaf. So, this any of these tissues, say leaf, you can use it to make cell cultures. So, that is the first step will be it form callus. That means, from the differentiated tissue you can make undifferentiated mass of cells, which is called callus. So, callus means they are neither root, neither shoot, neither stem, but a mass of cells. So, these callus may be often hard, but you can make it friable by manipulating the medium condition, and the from friable callus it is possible to raise cell suspension culture. And redifferentiation means, once you have the callus cultures, it is also possible from there to regenerate into plants, that is called redifferentiation. So, what is showing here in the left side, that is a callus, and callus if you treat with proper hormones, here it is written cytokinin, you will get shoots emerging out from the callus. Also, if you treat with another set of hormones, say auxin, low concentration of auxins, you may get root formation, and eventually whole plants can be produced. Now, what I said, it is very important that what you produce in the culture medium, so that the plants will grow, say it is for the callus or say it is for the shoots. Apart from the basic macro and micronutrients, you need to add vitamins and you need to add sugars, and also you need to add hormones. So, synthetic plant growth regulators are available. In addition to that, sometime tender coconut milk is used as one of the nutrients in the plant tissue culture, so that promote growth. Why? Because tender coconut milk contains cytokinin. Okay. And the third one is what is showing that it is a liquid culture, where bamboo plants are growing and it is just about to start its flowering. So, that means it is also possible to induce flowering in the culture condition. So, how to raise suspension culture? So, you have to take a plant material, you have to do surface sterilization, then put it in a proper medium containing hormones. So, from the leaf tissue it will form the undifferentiated mass of cells, which is called callus, which is called dedifferentiation. So, these callus can be made friable and these callus can be maintained in the appropriate medium, and from there it is possible to disintegrate the callus in the liquid culture, and then you can grow them in the liquid under shaking condition, and eventually you will be able to land up with the fine cell suspension. And these cell suspension cultures are indeed a very good system for inducing the plant metabolites. So, what is shown here is basically a fully grown plant cell suspension, so once it is grown for say 21 days or so, so it is kept under this condition. So, it is possible to measure the cell volume from this. So, why I am showing this is to just to give you an idea how it looks once suspension cultures are fully grown and ready for the subculture or ready for harvest for studying the metabolites. So, this is another recent example that even the coca cell suspension cultures have been raised and they are being explored as a source for the fragrance to be used in chocolate making. Now, there are some examples of the plant based metabolites which can only be detected in the cell culture, but not in the whole plant. That is very interesting. So, like in Kalmeg Andrographis paniculata, you will get paniculide, then podophyllum, that is an anti-cancer plant, it produces podoverin. Now, why it is produced, that is difficult to explain, but what what is important here, that cell culture actually the proper life cycle is not there, it is compressed. So, under that condition maybe a set of genes are expressed which are not expressing in the differentiated culture, and as a result of that you get some of these metabolites. But this is relatively rare, and now if you see in the left side that there are plant cell culture based products which have gone to the industry, that means they are produced at the commercial scale. One is seconin, one is gingosides, another is parpurine, and the Taxol is also another candidate which I have not put in the list. So, Taxol is basically the anti-cancer drug. Now, scientists actually they work for years after years, and they are able to develop high producing cell lines, and these cell cultures were capable of producing the desired metabolites of interest. But what happens often that once you get the desired metabolite of interest, that cell line you maintain, you subculture and maintain, then over the time you will find that there will be decline in the metabolite level. So, that means there is a problem of instability. So, cell cultures are not always stable. So, that is why there is always a need to screen for highly productive cell lines, which in turn is of course an expensive business for the industry, unless the product has a very high value and very very expensive. So, here what I have shown here is that the example of some of the specialized metabolites which are being produced in plant cell and tissue culture, and the name of the metabolites and the species have given, and the left side the chemical class like alkaloids, steroids, phenylpropanoids under which these metabolites belong have also been shown. It is also interesting to tell you that the essential oils can also be produced in the cell cultures. So, you all know that mentha is a good source of essential oils, lavender, eucalyptus. So, scientists in the past they develop cell cultures, and from cell cultures they are able to detect the metabolites. Not only the cell culture, they have also regenerated the shoots from the callus, and those shoots from those shoots they were able to detect the metabolites. That is indeed interesting. But what happens that again over the time the productivity gone down. So, that is one is the inherent problem. Now, if we look from a positive point of view, as I have said about the seconin. So, before that I ask you to concentrate on the left side. What is showing here? This is a growth dissociated product. That means, first the cell culture start growing, it goes to the log phase and then late log phase and stationary phase, and once cell culture attains certain stage of growth, then only the metabolites started synthesizing. So, that is called good growth dissociated product. That means, when you start growing the cell culture, you may not be able to get the desired compound. So, this strategy has been exploited for the production of seconin, the red dye from the plant Lithosphaermum erythrorhizon, and what I said before that this is the first plant cell culture based product available in the market, and still it is available in the market, and in the Japan the seconin is only produced by plant cell culture. So, based on this graph, what if you see here. So, basically this is nothing but a schematic diagram of industrial scale production of seconin. So, there are two tanks, one is 200 liter and one is around 750 liters. So, first one is basically for growing the plant cells in a particular medium. At that medium plant cell grows very fast, and so once it is grown, then basically the medium is being removed and the cells are transferred to another chamber where these are grown in another medium which is called M9 medium. Now, first one is the growth medium and second one is the productivity medium. So, the exact compositions are not disclosed, but normally if you can remove the nitrogen and phosphate sources, that means that gives some stress to the plant cell cultures, then that basically start synthesizing the desired metabolites of interest, as has been found also with carrot cell lines for production of anthocyanin. So, in the M9 medium there is certain deficiency of the certain macronutrients, and as a result of that M9 medium started synthesizing the seconin pigment. Okay. And in the next one, this is what is the original plant Lithospermum, and first one in the right side is basically the cell cultures grown in the growth medium, and second one cell cultures when they are transferred in the production medium, that produces the red colored dye, and this is the laboratory scale upliftment, that is the scale up at the laboratory level, and the right side bottom is basically the industrial scale production, which is being followed by Mitsui Petrochemical Industries for production of seconin. Now, apart from metabolites, it is also possible to produce the recombinant proteins in cell suspension cultures. So, one way is that from the tissue you develop the plant, or you can transform the plant with the desired gene, and then that gene is expressed from there. You can the transgenic plants can be used as a explant to make dedifferentiated cultures, that is the callus culture again. So, from there you can raise the suspension, and from these suspensions can be grown at large scale and then in bioreactor. So, the desired metabolites here is basically the proteins can be produced, or means recombinant proteins, and then later that can be processed, can be checked through the downstream processing, and eventually the products available as a finished product. This is a schematic, so it is also possible. Now, let us see about another aspect of tissue culture, that means that so far we have talked about the cell culture. Now, it is also possible what I said in the on the preview in the very first slide, subject covered, that differentiated culture. So, differentiated culture means these are not the cell suspension, but the culture either shoot or root. So, one important point is that when you try to make tissue culture, so you have to visit the field, collect the plant, and then bring it back to the laboratory and develop the culture. So, this is one such plant, Carnscura decussata, which is a threatened, and it produces bioactive xanthone compounds, which gives which are used for the treatment of nervous ailments. So, here in the laboratory the successful multi-port shoot cultures are produced, and not only that, from the proper propagation protocol was standardized, and then the plants were transferred into the field and they survived well. And also these plants can be maintained in a specific medium, so that the growth can be slowed down. So, you can keep it for the long term in the laboratory, as and when required you can make them growing faster by transferring into the other medium. So, apart from that, there is another plant I mentioned here, which is called Hopia fastigata. This is also very rare plant, and it has a sort of very short life cycle of maybe 1.5 months or maximum 2 months, even less than that. But these plants were known to produce bioactive xanthones as reported by the organic chemist. So, the plants were collected from a forest, and then attempts were made to develop tissue cultures, and with a very hard effort finally scientists were successful in producing these shoot cultures. And these shoot cultures, when analyzed, it was found that these shoot cultures are producing the xanthone metabolites, which are originally found in the field grown plants. So, this is basically a zoom view of the shoot cultures. Now, what I have said about the essential oil, the cell cultures and the shoot cultures derived from them can produce essential oil. So, where this is another species of mentha citrata, where people attempted to make axenic shoot cultures, and these shoot cultures, when they have analyzed under electron microscope, so they are able to find the epidermal glands which basically accumulate the terpene metabolites, and they have also analyzed the metabolites both from the parent plant as well as the shoot culture. And what you see, the principal metabolite which mentioned in this peak and here, which is mentioned here, they are the same. So, that means the shoot cultures grown in the laboratory are capable of producing the same or similar metabolite or metabolites provided you give the proper condition. Now, given such success from the cell culture, scientists attempted to grow the multiple shoot cultures in large scale for large scale production in bioreactors. So, these are the different laboratory scale bioreactors which are used for propagating the multiple shoot cultures at a faster rate, so that you can harness the desired metabolites of interest. And this is a pictorial diagram of different bioreactors which are used for growing the multiple shoots as well as for cell suspension cultures. And Korea particularly Koreans were very successful in developing bioreactor based propagation of plants, somatic embryos and so on. Now, apart from shoot, roots are the important source of metabolites. So, one can harness the metabolite from the root. So, the question naturally comes, is it possible to develop root culture from the plant? So, yes, it is possible, and in fact, long back in the last century, Professor H. E. Street first shown that the isolated root cultures can be grown in the laboratory, particularly the species he had shown is tomato. So, isolated root cultures can be grown in the laboratory provided you give the proper hormonal treatment. So, the same Canskora diffusa was used for inducing the roots, and with the proper hormonal combination it was possible to initiate roots from the leaf tissues, and later these roots were subcultured in the liquid medium, and then they were grown, and then different hormones were tested like different auxins, particularly like IAA, IBA, NAA, and the phenotypic growth of the root showed difference, and not only that, it also affect the level of the metabolites detected from there. So, here is another example that how from the adventitious root cultures proper root adventitious root initiation, proper root cultures are maintained, and then root cultures can be grown at large scale, and the metabolites such as plumbagine can be extracted from Prangabo species which was published. So, hairy roots, next come to hairy roots. What is the difference between normal root and hairy root? Okay, that I will tell. So, normal root cultures is that you develop the root lines from the plant tissue by hormonal treatment. So, hairy roots are basically these are called natural genetically engineered roots. That means, when dicot plants in particular are infected by a soil bacterium called Agrobacterium rhizogenes. So, what happens as a result of infection, a part of its plasmid DNA got transferred into the plant genome through the wounding site, and once the part of the plasmid DNA integrated into the plant genome, it alters the metabolism of the plant cells, and as a result of that, when metabolism is altered, as a result of that more auxin is produced, and when more auxin is produced, then we will see as a result of phenotypic manifestation is that roots coming out from the wounding site. So, the detail of the hairy root induction I will tell in the subsequent slides, about the detail about the molecular mechanism I will discuss later. So, here there are some examples of the plants and which produces specialized metabolites, and hairy roots are capable of producing all these metabolites as shown by the scientists earlier. So, here is a typical stepwise diagram how hairy root are induced. So, normally a leaf tissue is taken and the midrib is the best part where you make the wounding, and then there you inject the pure Agrobacterium rhizogenes log phase culture, and then you put it for the incubation, and after several weeks what you will find from the wounding site the hairy roots started emerging. So, these hairy roots later you cut it along with a little bit of mother tissue, you put it in the liquid medium. Once it started growing, then once you subculture after several passages, mother explant can be removed and hairy roots can be grown fully. And in order to kill the agrobacterium, sometimes the antibiotics such as ampicillin or carbonicillin is added, and after several passage totally hairy roots will be free of agrobacterium and it can grow, and these hairy roots basically reflect the metabolism what is occurring in the normal root. So, so that the metabolites which are produced in the normal roots can be produced in the hairy roots. But then one important point is this, that hairy roots basically it is itself the source and it is itself the sink, whereas if you consider the whole plant, it is basically the metabolites are synthesized in the root there, and the few metabolites are retained there, and there are other group of metabolites which are transported into the leaf. So, but in the hairy root there is no shoot. So, itself is the shoot, itself is the source, itself is the sink. So, the metabolites which are produced in the hairy root, they will accumulate there, and that is why when you analyze the metabolite content from the hairy roots as well as from the normal roots, you will always find more metabolites in the hairy root, and then the reason behind this is basically the source sink issue, and this is basically the induction of hairy roots of carrot. And these carrots hairy root cultures were capable of producing benzenoid metabolites. Now, in addition to normal hairy root, it is also possible to develop phototropic and photomixotropic hairy root by putting the carbon level, the sugar level minimum or totally avoiding sugar, and instead you provide them enriched carbon dioxide. These photosynthetically grown hairy roots, people have explored whether they are capable of producing some of the metabolites which are synthesized only in the aerial part or in the green tissue, and in fact, the results were indeed promising. Now, we have come almost to the last part of it. Now, what I have mentioned about the cell cultures, callus cultures, cell cultures, multiple shoot cultures, normal root cultures and hairy root cultures. So, these cultures can be modulated, these cultures can be treated by different empirical approaches, so that the metabolism can be modulated, and as a result of that you may get the metabolites at higher level. For example, you can manipulate the carbohydrate source, you can also make changes in the nitrogen source, you can make changes in the phosphate source. As I have mentioned before, that if you can make either phosphate or nitrogen limitation, so the cultures often produce the pigmented metabolites like anthocyanins. Even hormones levels can be manipulated, even light plays important role. By manipulating light, it is possible to synthesize the metabolites. Same is the case for temperature, osmotic stress, precursors. So, you can if you know the desired metabolites and if you add the precursor which should be cheap. So, then you can get the metabolite produced at higher amount. And finally, the elicitation, that means the cultures you can treat with some foreign molecules. So, these molecules could be the sources from microorganisms, say fungal cultures, it could be some hormones like methyl jasmonate, it could be something called chitosan which is obtained from the chitin, okay, and then it could be certain other like salts, like heavy metals. So, these can be added to the plant cell cultures, and when you add those things, of course, you have to add these under sterile conditions. So, you have to make sure whatever you are adding there, that must be sterile, and once you add this in the cell culture or root cultures, what plants roots or cells will think that they are under attack, because the cells or roots will treat them as foreign molecules. So, they are under attack, and then they will induce the defense response, and once the defense response is induced, that basically leads to the production of your specialized metabolites, as I have shown in the previous classes, that how the defense compounds are induced. So, specialized metabolites are produced by the plants to defend themselves. So, that strategy has been used by the scientists by using elicitor, and they were very successful in producing the metabolites at higher level. And so, in the next class basically we are going to discuss more about that. So, what in conclude, what is important, that indeed cell culture is an attractive way for producing metabolites, but one has to consider the capital cost and the cost of growth medium. So, that sometimes hindered the progress for commercially extracted compounds. And in terms of medium term, the future of cell cultures may be used mostly for biotransformation or to explore new products from cell cultures which is not available in the normal plants. And in the longer term, it may be possible to develop cell and organ cultures where a pathway genes can be expressed under the control of a regulatory gene, and once that is achieved, then sustainable commercial production of specialized metabolites from cell cultures and organ cultures will become more reality. With this, I end this class. Thank you.